Janet was trained in Cambridge and Oxford. That was where she did her graduate work. And then she was recruited to Canada by a young man who's her husband. And she was at the University of Brock University in Ontario for a number of years. So much of her work was reported from Samuel Lunenfeld Institute. And so through the years, every time there would be some next, particularly mouse embryo development that needed to be done, in a very short period of time, Janet would publish it. So she is an absolute mainstay in this field, and in the last few years has now begun to really drill down into stem cell lineages, embryo stem cell lineages, and has done some wonderful work, and was recruited to the Hospital for Sick Children in 2005, and is there heading up. She is now Chief of Research at the Hospital for Sick Children. It's quite a large research endeavor, and maybe she'll tell us just a tiny bit about that. But she's here today to talk to us about manipulating the mouse embryo from lineages to stem cells and back again. Thanks, Janet.
Thanks, Anne. Thanks, Anne. It really is a pleasure to be here. Anne has asked me before, and I've never been able to come, so I said, please invite me again. I will come. I think these kind of workshops where it's just a small group where you can really interact are always a lot of fun, and they do sort of open up new challenges. This is my favorite stage of development in the mouse. It's the blastocyst, and at this stage the mouse embryo is just about to implant in the uterus, and it has developed 3 distinct cell types. And that's all, only 3 cell types, so that's all it's done. It's taken 3 or 4 days to get to this point, so it's a slow process, and it's an initial stage of lineage segregation. And what we've been interested in over the years are the processes that set up those first 3 cell lineages in the blastocyst. And that's a fundamental question in cell fate and cell determination in development, but it's also important because it's from this stage of development, the blastocyst, that we can derive stem cells that can be extremely useful, like embryonic stem cells, but also other stem cells that reflect the lineages of the blastocyst itself. So what I want to do today is to give you a little bit of a story about what we know now about the lineages of the blastocyst, about the stem cells that arise from them, and if I have time, and I'm looking at the clock, I want to turn back at the end to the relevance of the mouse to understanding human development and human embryonic stem cells. So this is going to be almost entirely a mouse-centered talk because that's the system we know the most about, but I really want us to think about, and maybe that's a discussion for the day, about how we translate what we find in the mouse into the human system. So I need 3 hands. What we know from research over many, many years is that the 3 cell types we see in the blastocyst are the distinct progenitors of specific lineages in later development. So the blastocyst consists of an outer cell layer called the trophectoderm, which gives rise to the trophoblast layers of the placenta, and only the trophoblast layer. So a very important tissue for the survival of the mammalian embryo in the uterus. At one end of this cavity, the blastocele, is the inner cell mass, and by the time I've shown you here, about 4.5 days, the inner cell mass has segregated into 2 populations, a layer of cells on the surface called primitive endoderm, which, despite their name, endoderm, largely give rise to extra embryonic endoderms of the yolk sac and don't give rise in any major way to the definitive endoderm of the fetus itself. And it's these enclosed cells here stained in pink because they're stained with an antibody to OCT4, the famous pluripotency factor. It's these cells, the epiblast cells of the embryo, that are the pluripotent cells and that give rise to the entire fetus as well as some extra embryonic mesoderm tissues. So the mouse embryo spends its first 4 days making cell types, the primitive endoderm and the trophectoderm, which you throw away at the end of pregnancy and don't make a mouse. But those tissues are absolutely critical for the embryo to develop. They are required for a mammalian embryo to survive in the uterus. There's a famous saying by Lewis Wolpert about the most important time in your life is not birth, marriage, or death, it's gastrulation. I would argue that actually the most important time in your life is blastocyst formation and formation of the trophoblast, because if you don't have a placenta, you can gastrulate all you want, and you're not going to make it through pregnancy. And actually, one of the things that's an aside, as people have gone and knocking out genes involved in fetal development and adult development, in many cases they're finding placental phenotype. So the placenta is very susceptible to genetic disruption, so it's a very important tissue. So not only are they important for surviving in the uterus, those tissues, the trophectoderm and the primitive endoderm, are also key for the development of the fetus, because they are signaling centers that provide the initial signals that pattern the fetus itself into the anterior, posterior, dorsoventral, left-right patterning signals. And you've heard, I think, from Jaime Rivera before, and his lab have posters here, how important those tissues are for patterning the embryo. So we need to understand all three cell types in order to understand how the fetus develops. And if you were a human embryo at this stage, you would look very similar. You wouldn't look as beautiful. Human embryos are kind of scraggly. Mouse embryos are beautiful. But by the time a human embryo implants, which is a bit later than the mouse, it would look quite similar to this and would develop the same three cell types. So the initial phases, timing is different, we'll come back to it, but the three lineages are much the same. So three distinct lineages, and we can show, if we take the cells out, that they really are committed. They've made a decision, and cell fate is restricted. What we also know is that we can, from those three cell lineages of the blastocyst, derive in the mouse three distinct stem cell or progenitor cell lines that can be grown indefinitely in culture and self-renew and retain the restricted potential of the lineages from whence they came. And those are, of course, the most famous ones are the embryonic stem cells, which I mentioned, from the pluripotent cells of the epiblast. They express and require specific transcription factors that promote pluripotency. And if you put them back into the fetus, they will recapitulate the lineage development of the epiblast itself and contribute to the fetus, but not to the placenta and not to the primitive endoderm of the yolk sac. Several years ago, we showed you could derive trophoblast stem cells from the trophectoderm. They require their own transcription factors, and we'll come back to that in a minute. And when they are put back in the blastocyst, they contribute to the placenta and not the fetus. And then finally, we have these Zen cells that we derive somewhat later that have, again, distinct phenotype, distinct transcription factor requirements, and contribute only to the yolk sac. So a very nice system then, where we can use these self-renewing cells to look at molecular pathways, go back to the embryo and ask how they work, etc., etc. So that's really what I want to do today, is to talk about the issue of how lineage fate is restricted in both embryos and stem cells. And we're really going to be focusing largely on those transcription factors and their regulation. So how are these lineage-specific transcription factors regulated so that they can restrict cell fate? So I talked about the trophoblast, and if you saw on that slide, one of the key transcription factors for the trophoblast is a transcription factor called CDX2. And CDX2 turns out to be both necessary and sufficient for promoting trophectoderm and trophoblast development. And a number of experiments have indicated that. In particular, you can, although embryonic stem cells and TS cells grown in normal conditions are restricted in their fate, you can actually switch cell fate from one to the other. And in particular, you can take ES cells and turn them into trophoblast stem cells by altering transcription factors. You can either take away OCT4, which causes the upregulation of these trophoblast transcription factors, or you can ectopically express CDX2 itself, which causes the pluripotency genes to be downregulated and TS cells to come up. If you do that, the cells will differentiate into differentiated trophoblast cells. You have to change the culture conditions to the growth factor requirements for TS cells, and then you can go from ES cells to TS cells. And this was work initially done by Hitoshi Niwa, and then in a collaboration with our lab, the work on CDX2. So CDX2 alone can drive fate from pluripotency to trophoblast. So what happens if you take it away? Well, if you take it away in embryos, and this is I should just say, if we go back to the embryo, CDX2 expression is a key anticipatory expression of the trophectoderm lineage. So if you look at CDX2 expression in the early blastocyst, it's expressed specifically in the outside cells, and even in the late morula, as you'll see later, it's in the outside cells. OCT4, the pluripotency gene, these are antibodies in both cases, the protein is still expressed, and the message in fact, in all the cells of the early blastocyst. So CDX2 is restricted before OCT4 is restricted. So CDX2 is still the earliest restricted lineage transcription factor we can see in the mouse embryo. So it comes on first. Does it play a role then in really promoting trophectoderm and perhaps downregulating the pluripotency genes to initiate this lineage segregation? So what happens when you take it away? And this is old stuff, so I'm only going to go through it very quickly. If you take away CDX2 from a normal embryo, then what happens is the embryo will get to the early blastocyst stage and then kind of collapse. It cannot maintain or differentiate the outer trophectoderm. None of the differentiated markers come on, and the trophectoderm cells will apoptose. The embryo will not implant at all. So this is a pre-implantation lethal phenotype. But it does begin to form a blastocyst. However, when you look at that blastocyst, where you start to see restriction of the pluripotency genes to the inside cells, in a CDX2 mutant, they remain ectopically expressed in the outside cells, suggesting then that CDX2 is absolutely required to maintain trophectoderm and to downregulate the pluripotency genes. So CDX2 is necessary and sufficient for specifying trophoblast cell fate in the blastocyst and in stem cells. It's necessary for downregulating pluripotency genes in the embryo, and it is necessary for promoting implantation. So you really don't get functional trophectoderm. But you'll have noted in the diagram, and I'll show you pictures in a minute, that CDX2 embryos do begin to initiate formation of blastocysts. So there's this nice gene expressed early, it does all these great things. You'd expect maybe the embryo just to block before you even begin to form the blastocyst. That's not the case. Suggesting then that there may still be things going on, of course, upstream of CDX2. So in the embryo itself, are there other genes involved in initiating the trophectoderm pathway? And what we know now is that that's the case, that CDX2 is downstream of another pair of transcription factors, TEAD4 and its co-activator YAP. So this was work from two groups, Melda Pamphlis' group and Hiro Sasaki's group in Japan, who knocked out another transcription factor, TAD4. It belongs to a gene family that I'll come back to in a minute. They certainly weren't looking for pre-implantation, at least Hiro wasn't. But what they found was that these embryos looked quite similar to CDX2 mutants. They failed to hatch, they failed to implant, and they also, like CDX2 mutants, failed to downregulate pluripotency genes. But in a TEAD4 mutant, you got no CDX2 expression, and you really seem to have a more severe phenotype than the CDX2 alone. So what this suggests then is that TAD4, this transcription factor, is actually upstream of CDX2, and the fact that the phenotype is more severe suggests that there are other genes downstream of TEAD4 that are also required for the next phases of trophoblast development. And again, without going through a lot of published work, we now know that that's the case, that TEAD4 sits currently as the transcription factor at the top of the trophectoderm hierarchy. Downstream are at least two transcription factors acting in parallel, CDX2 and GADA3. CDX2 has this additional role of downregulating the pluripotency genes, and CDX2 is autoregulatory. We know a lot, of course, about the pluripotency modules from studying ES cells, and this is a highly simplified version, but what we know there too is that once they're established, there are autoregulatory feedbacks that maintain these pathways. We also know that OCT4 itself can downregulate CDX2, so we have an interesting situation here of a reciprocal negative interaction here and an autoregulatory loop, which, if you think about it, is the way you need to establish and maintain lineage segregation. So this is the lineage segregation part, but there are other players that cause the trophoblast differentiation pathway downstream. So TEAD4 sits at the top, and CDX2 is a key player in this sort of lineage segregation. But there's a secret that I didn't tell you about TEAD4. I showed you how CDX2 is beautifully restricted to the outside cells, and if that's what's going to drive trophoblast development, you might expect TEAD4 to also be restricted to the outside cells in the embryo, and that's not the case. GATA3 is, I should say, but TEAD4 is not. So it's expressed everywhere throughout the pre-implantation embryo. So how can this be working? How can you get a very specific effect on the trophectoderm lineage? Well, remember I told you that TEAD4 doesn't act alone. It's a transcription factor that requires a co-activator, YAP. And what I'm going to show you is that the pathway, it's not TEAD4 whose expression is regulated, it's the expression of the co-activator, YAP. And this takes us into thinking a little bit about how we would set up a difference between the inside and outside cells during the early development that could lead to this segregation of the two lineages. So this is a quick reminder of many years of work at the cellular level studying pre-implantation development to show you that there are important cellular events that precede blastocyst formation. That as you start at the 8-cell stage, the embryo undergoes a process called compaction, whereby these cells become, instead of being rounded, separated cells, they start to polarize, and you can't really tell the distinctions between them. You get adherence junctions, and eventually you get a proper apical basal polarity with all your favorite apical basal polarity genes being segregated. Between the 8 and 16 and 16 to 32 cell stage, there are 2 rounds of cell divisions in which the outer cells retain this polarized phenotype, but they bud off apolar inside cells, so that the polarized cells form this polarized epithelium, which will go on to form the trophectoderm, and the inside cells, the apolar cells, are trapped by this epithelium and go on to form the inner cell mass. So if you think about it, this process of forming an epithelium, generating an inside environment, is a perfect one in which one can start to think about, are there actually signaling pathways that are differentially active in the inside and outside cells? Concomitant with this process, you see the segregation of expression of genes like CDX2. It starts sort of all over the place at the 8-cell stage, and then as you go through this establishment of the polarized epithelium, we get this down-regulation of CDX2 in the inside cells and the upregulation in the outside until you get to this blastocyst stage. So it's the expression is concomitant with this process of compaction and polarization. So generating inside environment, are there signaling pathways that are differentially active in the inside and outside cells? And what's this got to do with TEAD4 and YAP? Well, turns out that there is a very important signaling pathway that is differentially active in the inside and outside cells in the early embryo. And this is a pathway that has really one of those rare signaling pathways that we didn't know about maybe 10 years ago, and all of a sudden, practically every paper you look at has something to do with this signaling pathway. And this is the Hippo signaling pathway. And the Hippo signaling pathway was originally identified as a growth control pathway in Drosophila, and then very rapidly, actually, it was shown that this pathway is highly conserved into mammalian cells. And since it's a growth control pathway, mutations in this pathway are really involved in tumorigenesis. So this is an oncogene type pathway, and it's involved very heavily in a number of different tumorigenesis situations, and indeed in normal growth control situations. The interesting thing about the Hippo pathway, well, first of all, we still don't know all the components, so it's an open field, but the core components are shown here. And this is the sort of Drosophila side of the equation. I've put them all together. This is both Drosophila and mammalian cells. This is the situation where the pathway is active, and this is the situation where the pathway is inactive. And the core parts of this Hippo signaling is this complex of proteins that contain a serine-threonine kinase called Warts in Drosophila and Latz kinase in mammalian cells. And when this kinase is activated by an external Hippo-activating signal that's related to cell-cell contact and cell adhesion and cell signaling, this Hippo-activating signal, whatever it is, and we'll come back, activates this complex. And what this does is phosphorylate this co-activator YAP, which is called Yorkie in Drosophila. When it's phosphorylated, YAP is kept out of the nucleus. It binds to the scaffold protein. It cannot enter the nucleus and activate downstream transcription. So TEAD4, or scalloped in Drosophila, sits on its targets, needs the co-activator. When Hippo is active, downstream transcription is inactive. And I know that's sort of backwards and it always makes your brain hurt a bit, but I want you to try and remember that. So when Hippo is active, the downstream pathways are inactive. When Hippo is not activated and there's no upstream signal, this complex falls apart, the kinase is inactive, your key enters the nucleus, and downstream transcription occurs. So here we have TEAD4 expressed everywhere. You might predict then that if this is a key activating pathway for trophectoderm formation, then what we need to look at is the nuclear distribution of YAP. So it had not escaped our attention, and Amy Ralston, when she was a postdoc in my lab, had already got antibodies to YAP and was beginning to look at this. And then we started to exchange information with Hiro Suzaki's lab at CDB Recon, who are also pursuing this. And together we published a paper in Developmental Cell a couple of years ago, which I will say right off the bat that they did the majority of the work, but we contributed both in the initial stages and some of the later analysis. But the story is really neat. So if you look at YAP, here is antibody to YAP. And you see it's sort of cytoplasmic and nuclear in early development. And then as we go through the 8 to the 16 cell stage, and you look at CDX2 expression, what you see is that all of a sudden YAP starts to become nuclear localized in the outside cells and actually actively excluded from the nuclei in the inside cells. And this is correlating very nicely with the upregulation and downregulation of CDX2 in the inside and outside cells. As you merge them here. So that was the first piece of information. So then what Hiro's lab then wanted to do was a series of manipulations in the early embryo to show that this nuclear localization was actually controlled by the LAX kinase. And here's just just a selection of some of the experiments they did, and we'll see some of these from my own lab later on. And what they showed here, for example, they overexpress the LAX kinase, so now you're going to activate Hippo signaling everywhere. It's going to be activated in outside and inside cells. If you activate it, you're going to turn off CDX2 because TEAD is going to be inactive, and that's what you see. You see that you don't get CDX2 expression. If we put in a kinase dead LAX2, then what you should be seeing is that you can now no longer activate the Hippo signaling, and you're going to get ectopic CDX2 in the inside cells, and that's what you see. And then here, they put in a non-phosphorylatable form of YAP, which should be able to enter the nucleus at all times. It's not phosphorylated by the kinase. And again, you see this ectopic CDX2 in the inside cells. Series of experiments, all suggesting then that indeed this pathway, the Hippo signaling pathway, was really key to really initiating the events that are going to lead on to the segregation of inner cell mass in trophectoderm. That Hippo signaling is activated in the inside cells, so we start with this sort of random expression, that as we establish an inside environment, Hippo signaling is active in the inside but not the outside cells. Therefore, YAP is phosphorylated, excluded. In the outside cells, there's no Hippo signaling active. YAP enters the nucleus, CDX2 expression occurs, downregulate OCT4, and we're off to the races. Lots of questions remain, but it's very nice because this is the first situation where we have a post-translational event initiating a transcriptional downstream events, which is what you have to initiate a developmental process. The questions that remain are pretty obvious, and certainly one of them is what's upstream of the LAX kinase in these inside cells. So Katie Coburn is a current graduate student in the lab, has been exploring this in manipulating these pathways in the embryo. And so she looked at what we know about the upstream components of the Hippo signaling in mammalian systems particularly. And what was known is that upstream of this complex, there are these two molecules, firm domain proteins, and NF2, or Merlin as it's called in Drosophila. NF2 is a tumor suppressor gene, so mutations in this cause form of neurofibromatosis. But both of these genes are known to activate the downstream path, the LAX kinase complex. She tested a number of components of the complex itself to see if she could find their involvement, and then she tested in particular the involvement of NF2. So what I'm going to show you is that Merlin seems to be very important in the pre-implantation embryo upstream of the LAX kinase. And the way she did this was to look at what happens if you put in a dominant negative form of NF2. So if NF2 is required for the activation of this pathway, if we knock it out, then what you would expect is that the LAX kinase will be inactive, YAP will be phosphorylated, and basically we're going to have an inactivation of the Hippo signaling pathway. So what you see here this is just controls where she's injecting GFP. In this one, she replicated the Sasaki experiments, putting in the LATS kinase dead form. And what you see there is that you start to see ectopic CDX2 in the inside cells. If you put in the dominant negative NF2, we get the same effect, in fact, even stronger. So if you block NF2, then you really seem to block this whole pathway and you get ectopic CDX2 expression. Now that's great, but you could argue, well, maybe NF2 is activating CDX2 by a different pathway. How do we know it's actually acting through the Hippo signaling pathway? Well, to address that, oh, sorry, missed out a part here. Well, I guess this is one part of it. If it's acting through the Hippo signaling pathway, then we should see changes in the phosphorylation of YAP concomitant with the blocking of NF2. So here we have the dominant negative NF2. It's hard to see, these are not easy, but the phosphorylated form of YAP is in the inside cells here. And when we look at the dominant negative, you see no phosphorylated YAP in the inside cells. So suggesting then that it really is acting through the Hippo signaling pathway. And then more importantly, she did this experiment here where she asked whether she could rescue the dominant negative form by activating Hippo signaling downstream. And she did that by injecting in excess of the kinase. So now we lack NF2, but we should be able to bypass that if it's acting through this pathway. And she did it this way where she made a sort of chimeric situation, knocked down NF2 in both cells with two-cell embryo, and then injected one of them only with a LATS that was tagged with GFP. So then you can follow the cells that have got activated YAP and compare them with the cells that have dominant negative NF2. And what you see is shown here, that if you look at the situation, essentially the green cells are the ones that have the excess LATS, and then here we're looking at YAP and its nuclear localization. So just look at these inside cells circled here. This cell here only had the dominant negative NF2, and it has activated YAP in the nucleus. These cells have got dominant negative NF2 and then the LATS kinase, and we see that we go back to the normal situation and YAP is excluded. Same thing down here. These are in this case, these cells over here have the LATS kinase, no YAP. This cell here does not, and it's got LATS in the nucleus. The arrows have got moved here, sorry. So basically then you can rescue the effects of Merlin by activating the downstream pathway. So that then suggests that we've put at least one more player in the story here. So NF2 is acting in these inside cells upstream of this to activate this pathway. So fine. Now, as ever, that's one more component, but we've still got a lot of questions, and I'm going to tell you right now, we don't know the answers to all these questions. But the question becomes really, how is Hippo signaling inactivated in the outside cells? The inside cells are in close cell-cell contact. That's very typical of any other Hippo signaling pathway. They have the opportunity to talk to each other. But the outside cells also have cell contact. Less of it, admittedly, but they don't seem to activate this pathway. So what is going on in the outside cells to inactivate Hippo signaling? And does this correlate in some way with the formation of this outer polarized epithelium? So what is the role of polarity and cell contact in activating Hippo signaling and causing this lineage segregation? So this is the kind of pathways that people are beginning to put together now, and about what's upstream of the sort of LATS kinase. Here's Merlin. And you'll see that activating Merlin does involve components that are involved in cell-cell adhesion, such as E-cadherin. So maybe E-cadherin activation is important for Merlin activation in the inside cells, and that really E-cadherin and cell adhesion is really what's regulating the process. So what happens if you take an embryo and get rid of E-cadherin? It's not a happy embryo. Sorry, Alison, I'm going to move. This is an embryo in which both maternal and zygotic E-cadherin have been knocked out. So there's no E-cadherin in these embryos. They look all right at the moment. But watch, this is the blastocyst. These are the E-cadherin mutants. They are not able to form a blastocyst. They continue to divide. As you can see, they have many, many cells, normal cell number, but they cannot form an organized blastocyst, and they don't form an organized polarized epithelium. So really and truly, in the absence of E-cadherin, you don't get proper segregation. But what happens then? What do these cells think they are when they can't sit together? Are they going to have activated Hippo signaling, or are they going to have inactivated Hippo signaling? So Robert Stevenson in the lab did a series of experiments. They were published, so I'm not going to go again through them in detail. Suffice it to say that when you do this and knock down, remove E-cadherin, this is the maternal zygotic over here, we see very few cells that express OCT4, and almost all the cells express CDX2, whether they're inside or outside. So in the absence of cell adhesion, all the cells actually express polarity markers. They kind of individually polarize, and they go on and express CDX2. And not shown here, they also have YAP in their nuclei. So cell adhesion, and you take that away, everybody thinks they're outside. So you do need cell adhesion to get the segregation of the inside-outside. But is it really related to just adhesion? Or what happens if you just block cell polarity without blocking cell adhesion? So Robert spent, these are unpublished, so maybe we could just stop here, sorry. Robert spent a series of experiments where he's really tried to address the involvement of cell polarity in this process and tried to block cell polarity without blocking cell adhesion and cell division. It's quite difficult to do, but he found some conditions in which, particularly using the ROCK inhibitor, so blocking Rho kinase, the embryos will continue to divide, and they sort of form these, morally, they never form a blastocyst. They continue to divide, but you can see they also have markers like LGL, which is a basolateral marker, all over the cell surface. And when you do that, these embryos think they're inside cells, they activate Hippo signaling, you see no nuclear YAP, and you see very little CDX2 expression. So polarity seems to be required to block Hippo signaling, and if you don't have polarity, even if you have cell adhesion, then you're in trouble. So he then did the other experiment and asked, well, what's the relationship between the two? Which one is more dominant? Is polarity just sort of secondary to the cell adhesion, or the other way around? And the experiment is shown here. And what we see is that even when cell adhesion is disrupted, when everybody should be outside, if you then block cell polarity, everybody thinks they're inside. So here, here's the blocking E-cadherin. We see a lot of CDX2 expression. Here's what happens when you block with the ROCK inhibitor. Everybody thinks they're inside, little CDX2. And these are E-cadherin mutants treated with a ROCK inhibitor. And they look just the same as the ROCK inhibitor. So even when you don't have cell adhesion, if you block with the ROCK inhibitor, everybody thinks they're inside and activates Hippo signaling. Is this really going through the Hippo signaling pathway? So Robert and Katie got together and did this experiment where they used the ROCK inhibitor and treated the embryos, as shown here. You get reduced CDX2 expression. They then, this is just a control with GFP, and then they took those and injected the kinase-dead lats. So now you're blocking Hippo signaling, and when you do that, you rescue CDX2 expression. So it appears that polarity is important in acting through the Hippo signaling pathway. So this is our current model. Many, many questions, don't have the answers. But what we think then is that there is a very specific linkage between cell polarity blocking the activation of Hippo signaling in these outside cells, allowing CDX2 expression. And in the inside cells, we see activation of the Hippo signaling through NF2. Also, in some work we did with Jeff Rana's lab involving the crumbs polarity proteins in the inside cells. And then you downregulate CDX2 and begin this segregation. So there's a lot of, obviously, important things to understand. How do you activate it upstream of Merlin here, and how do you inactivate it in the outside cells? Don't know the answers, but we're working on it. I think it's a neat system. It's the first case where actually Hippo signaling, which is doing everything, was one of the first examples of where it actually seems to be involved in a cell fate decision rather than specifically in a cell growth decision. But using the same modules, which is kind of what development does over and over again, use the same modules in different ways. So that's the first cell decision, making trophoblast versus inner cell mass. What about the next cell decision that takes place within the inner cell mass? to form the epiblast and the primitive endodome. The epiblast, now we're getting to the cells that most people really care about, the pluripotent cells, the ones that are going to make embryonic stem cells, all these extra embryonic cell types, you know. Nobody cares. They're very important, but people don't care in the same way. But you better care about this decision because this is key to really set aside the pluripotent cells from which stem cells arise. So how does it happen? Well, you saw in the pre-implantation stages and forming trophoblast and inner cell mass how dependent it was on cell position, cell polarity, cell adhesion, really forming this organized structure with an inside and outside cells through 2 rounds of polarized cell divisions. How does this decision take place? Well, we used to think it was the same because you saw the primitive endodome forming on the surface of the inner cell mass. So the idea was that, again, it was driven by cell position and maybe signaling differences between the inside and outside cells of the inner cell mass. We now know that it almost certainly is not the way it works. And the first sort of indication of that came when we just looked at the expression of the key lineage transcription factors within the inner cell mass rather than the stages we were looking at between the inner cell mass and the trophectodome. And this is a more advanced version of what we published several years ago now, as people have gone and looked at this in more detail. But if you have antibodies to CDX2, GATA6, which is a primitive endodome marker, or NANOG, which is a primitive epiblast marker, and this is a sort of 3.5-day blastocyst before you can see the primitive endodome. It's a flattened confocal image, so everybody's flat. But you can see CDX2 is in all the outside cells, nicely segregated. And in the inner cell mass, where everybody's expressing OCT4, there's already some interesting mosaicism going on in terms of expression of these lineage tracers. So there are cells that are GATA6 positive, there are cells that are NANOG positive, and there are cells still that seem to be expressing a little bit of both. So you've got an interesting mosaicism with no spatial patterning to it. This is totally random, apparently, within the inner cell mass. That's intriguing because it's also been shown in the last few years, and this is just a few of the many papers that have addressed these issues, that if you take just mouse ES cells and grow them in the sort of standard culture conditions, they also seem to have this kind of heterogeneity of gene expression in a sort of random pattern. There are fluctuations. They're actually going up and down in NANOG, stellar and REX1, sort of pluripotency markers, and then sort of complementary fluctuations, as you saw in that inner cell mass, in markers of differentiation, both mesoderm and indeed markers of extraembryonic endodome like GATA6. So ES cells are heterogeneous, and we know that this heterogeneity is controlled. Part of it may be stochastic, but it can be controlled by levels of FGF signaling. And that's shown, I think, in the next slide. There are many different experiments, again, that show the importance of FGF signaling, and particularly downstream of FGF, the ERK signaling pathway, in this heterogeneity in ES cells. And these are just the results from Frederick Lanner, who's a postdoc in my lab, in experiments he began when he was a graduate student in the Karolinska and completed in my lab, published a little while ago now. Where he was looking at the role of FGF signaling, but also the role of heparan sulfate proteoglycans, which are required with FGF to present it to the receptor. So what he was able to show, here's a typical picture that everyone would see if you stain ES cell colonies with markers like NANOG, KLF4, which are pluripotency OCT4 markers. You see they're not expressed in all the cells. But if you treat them with a chlorate, which actually inhibits heparan sulfate proteoglycans, then what you see is that all of a sudden almost all the cells express the pluripotency markers at 80%. The same thing if you block with an FGF receptor inhibitor, and then these are mutant cells that cannot produce heparan sulfate proteoglycans, and again everybody becomes becomes epiblast or ES-like. So you remove the heterogeneity by blocking FGF signaling. And that's just shown more clearly as you do the sort of individual levels here. You can see that everybody starts to really be consistently expressing the pluripotency markers. And if you do the same thing with the differentiation markers, you see them disappearing as you do this. So many experiments then suggesting FGF signaling is important for this heterogeneity. And that really there is a sort of dynamic ES cell state where there's a kind of flipping backwards and forwards around the FGF pathway in at least two different directions, towards mesodome and extraembryonic endodome. And that if you step out of this, get sort of over the hump, then you can go down and differentiate into the germ layers, or over a different hump, you can go and differentiate into primitive endodome. So it's a key component. Is FGF signaling then important in the inner cell mass? And is it true that maybe the inner cell mass is in this sort of reversible metastable state? And is it controlled by FGF signaling? Well, if there's a reversible metastable state in the inner cell mass, unlike ES cells, which seem to be able to do it consistently forever, in the inner cell mass, that's clearly not the case. If it's metastable, it's for a short period of time, and it rapidly stabilizes itself to establish the two lineages. And you can show that, for example, here. I'm going to move again. With a movie. Oops, sorry. In which what you're seeing here is a GFP tagged to the PDGF receptor alpha gene, which is a nice primitive endodome marker. And what you're seeing is it coming on sort of randomly within the inner cell mass. Then as the embryo grows over time, you see that cells either migrate to the surface, turn off the gene, or if they really end up in the wrong place, they will actually die. So finally, you end up with this, what we expect to see in the final embryo, is this very discrete surface of primitive endodome cells on the edge of the inner cell mass. This was first nicely shown by Berenika Pluser, and this was a movie actually made by Yojiro Yamanaka when he was in my lab. So the heterogeneous pattern is not there for a long time. It resolves over time. But is it dependent on FGF signaling? The answer to that is yes, absolutely. We first showed that several years ago now by looking at a GRAB2 mutant and showing that if you don't have the activation of the ERK pathway, then essentially you can't differentiate primitive endodome. And that's just shown also here. So again, work from my lab, Yojiro Yamanaka and Frederick Lanner, some work from Jenny Nichols' lab also showed this part of the story. That is to say that if you treat mouse embryos from the 8-cell stage with an FGF receptor inhibitor or an ERK inhibitor or both, then what happens is that the embryo, which should have this sort of mosaic pattern of GATA6 and NANOG, ends up with almost all the cells of the inner cell mass expressing NANOG, and only maybe one or two in this particular case expressing GATA6. So no FGF signaling, you've shifted everybody towards the epiblast pathway. Now you could argue that's just permissive, you need it to make primitive endodome. If this is really instructive and controlling this heterogeneity, then you should be able to shift the cells in the opposite direction. And that was the key experiment that we did, in which we treated then 8-cell embryos with FGF4 at high levels, and now where a control is mosaic, all the cells in the inner cell mass express GATA6 and nobody expresses NANOG. And actually they start to migrate on the surface of the inner cell mass, behaving like primitive endodome cells. Importantly, Frederick in the lab then went on to do a series of time course experiments to ask when is the embryo responding to FGF? When's the susceptible window? And it was a bit of a surprise, at least a surprise to me. That's probably just because I didn't think about it very hard. The surprise was that the time window of sensitivity was during the formation of the inner cell mass itself. So those experiments were grown from the 8-cell stage, so you don't know when it's happening. But in these experiments, he did a series of experiments in which he basically added inhibitor or added FGF at different times, then took it away. So if we look at the top one, he's growing in the FGF and ERK inhibitor. If you grow the embryos all the way through, right through to 4.5 days, then, and don't remove it, then essentially we see the situation where everybody becomes epiblast and NANOG positive. If we grow the embryos in the inhibitor to the early blastocyst or even mid-blastocyst stage, take the inhibitor away, we reconstitute the mosaicism. So even though the inner cell mass has begun to develop, take away the inhibitor, the embryos sort themselves out and become mosaic. And even at a sort of fully expanded blastocyst, the same thing is true. And it's not till you get to this stage that the embryo cannot go back. So we've restricted the lineage. The opposite is shown here. The embryos were grown in normal conditions all the way through, and you end up with a proper inner cell mass with epiblast and primitive endodome. But again, if you add the inhibitor, even after blastocyst formation has begun, you essentially inhibit the pathway, and everybody becomes epiblast. And then this is my favorite, because we have the two opposites at each end. If you grow them in FGF4 all the way through to E4.5, everybody's primitive endodome. If you grow them in FGF4, even up to the mid-blastocyst, and then take away the FGF and add the inhibitor, you flip everybody back the other way. And if you sort of do it at this stage, you kind of balance out and reconstitute a normal embryo. So it's very sensitive, and it's a middle window in the inner cell mass itself. So that's intriguing then, and it tells us that at that stage, somehow, the cells within the inner cell mass are reading out FGF levels, and they're reading this out such that eventually, where they're close and talking to each other with maybe a minor difference, one has more ligand, one has more receptor, eventually this minor difference segregates out into a major difference such that we get a separation of the two cell types. Downstream genes are turned on that affect cell adhesion, and the cells sort themselves out and become completely separated into the two lineages. So FGF signaling drives this gradual segregation. We don't know how it really begins, and that's a real question. We know from single-cell analysis from Michinori Saito and from Paul Robson's lab that in the inner cell mass there are differences between FGF4, reciprocal differences between ligand and receptor in the inner cell mass, explaining perhaps the beginning of this pathway, but it doesn't explain how the initial mosaicism arises. And it's still unknown how that mosaicism arises. Is it stochastic, or is it lineage related? So I want to just quickly, I'm looking at the clock here, there's a very nice hypothesis would suggest that it could be lineage related. I like this hypothesis. We're not sure it's true. There are 2 rounds of polarized cell divisions generating these inside cells at the 8 to 16, 16 to 32. And you could imagine that because the conditions in the outside cells are going to be different at each round, that those 2 rounds of cell division would generate epiblast progenitors and primitive endodome progenitors, and that's enough to initiate the mosaicism. We know it can't be a hardwired process because I just showed you that at the blastocyst stage we could change the fate of all the cells in the other direction, but it could be enough to sort of, in the normal situation, give them a kickstart. So Yojiro did a series of lineage tracing experiments, which I won't have time to go through. Suffice it to say that when we followed in the embryo itself the fate of the primary inner cells, the 8 to 16 cells, and the secondary inner cells, the ones that went in at the next stage, we couldn't really see any difference between their fate in the later embryo. So no clear predisposition. However, I should note that using a different lineage tracing system, Magdalena Zernicke-Gertz's lab has come to a different conclusion and does see some bias in the secondary cells towards the primitive endodome fate. So it could have some role, but it clearly isn't the only thing that can happen. The cells can change their fate in midstream. So we have an interesting system in the blastocyst, actually, where within the inner cell mass, we have a set of cells that are going to go on and form the epiblast cells that produce this signal, FGF4. Receiving cells next to them in the inner cell mass will respond by forming primitive endodome. And what I didn't tell you today was that the trophectodome cells also require FGF4. They don't require it for their cell fate, they require it for proliferation to form TS cells. And that signal then is coming from the same cells, and the trophectodome cells take the signal and proliferate, and the cells in the inner cell mass take the same signal and change fate. So it's a very parsimonious way of using a single signal from a single cell to do 2 different things in this stage of blastocyst formation. So it's really kind of a neat system, and we're beginning to obviously dig down into the details of how a mouse blastocyst is put together. So what I want to, oops, no, sorry, not sure I can go back. No, I can't go back. Come over and go back. So just to quickly finish up and say that what we know about lineage formation in the blastocyst also explains how we get 3 different stem cells from the embryo, because they all come out in different FGF conditions. We now know that ES cells like to grow in conditions where you inhibit FGF, not surprising, because I just told you that when you grow a blastocyst in FGF inhibition, everybody's going to become epiblast, and you'll get ES cells. You won't get the other cell types. If you take a blastocyst and grow it in FGF, the trophectoderm will proliferate, and of course the primitive endoderm will differentiate. So if FGF conditions promote Zen cells and TS cells, inhibiting FGF makes ES cells grow. So it all makes sense. But now, what happens if you think about a human embryo? So human blastocyst development and human blastocyst-derived stem cell lines, human ES cells are usually maintained in conditions that in the mouse would promote trophoblast stem cell proliferation. That's FGF and actually activin and nodal. If you grow them in FGF inhibitors, they are not happy at all. It's been shown that human ES cells are more similar to a different pluripotent cell type that arises from the later epiblast that is FGF dependent. And also, we've been able to show, or been unable to show, that we can derive human TS cells from human blastocysts. So, So how similar are mouse and human development? So all that information I told you about the mouse development, does it really relate to the human? Why is it that when we take human blastocysts, we get a different cell type, really, when you make a human ES cell? And we still don't know an awful lot, and that's one of my messages today, is we need to know more. We know that the timing is different, and I already explained that. And we also know that the expression of the different markers, CDX2, and I'll show you that, does come restricted to the outside, but it seems to stay on a little longer in all cells than it does in the mouse. And it's been shown that OCT4 stays on longer in the human trophoblast than it does in the mouse. And so not much longer, so by the time of sort of 6 to 7 days, you get this restriction, but the blastocyst gets much further without showing the segregation of the two cell types. So there's a timing difference. Is that timing more than timing? Is it functional? We don't know. Here, for example, is a picture from our lab where we used antibody to CDX2. This is a nice, beautiful, expanded human blastocyst. CDX2 is restricted to the outside. Here's the inner cell mass, and here's nanog in the inner cell mass, but actually still in some of the outside cells at this point. So the pluripotency genes seem to be less sensitive to the expression of the trophectoderm genes. And that's a situation you see in other mammalian species, as you've seen in the cow and other systems, a difference in timing. Is it fundamental? We don't know. So we also have to think about the fact that not only is the blastocyst timing difference, the post-implantation events are very different as well. So when the mouse embryo implants, the trophoblast here continues to proliferate and forms this extraembryonic endoderm. And it's from these cells that are in contact with the FGF signal from the embryo that we get TS cells. A human blastocyst implants, and the first trophoblast is actually invasive, moves away from the embryo, is not likely to respond to any FGF signal. And it's really not late until later where you get the chorionic villus formation that you get a proliferating trophoblast population. So that might explain then why we don't get TS cells. But also, the embryo itself doesn't form this weird rodent egg cylinder. It forms this disc that is much more similar to, a rabbit or any other system you might want to look at. And again, the spatial and geographical signal relationships are going to be different. So I think it's very important to remind ourselves, as we always have to as a mouse person, that a mouse is not a cow, as I said in the nature commentary, but it's also not a human. And we need to really follow and understand this in more detail. So I think that human blastocyst-derived trophoblast is unlikely, it's probably not responsive to FGF proliferative signals, therefore no TS cells. It really is not the same cell type. The issue, however, is really more important and more interesting is what happens in the inner cell mass. So is the inner cell mass sensitive to FGF signaling in the same way as the mouse is? And we're beginning to do some of those experiments, and Jenny Nichols in Austin Smith's lab is doing some experiments there. And we'll wait and see what the results are, she says, since this is being streamed live. But it's clearly an important question. I think it's very important then that we have a huge ability to manipulate the mouse embryo and understand the mouse embryo. And that's going to give us certainly guidelines into the initial events of lineage formation. I'd be surprised, extremely surprised, if the major transcription factor pathways are not similar. In fact, they do seem to be. But the details and the signaling interactions and the geographical and spatial interactions may be different. And so we do have to have more careful comparative analysis of pluripotent stem cell lines of all different sorts, and indeed of human and mouse embryos as well. So I will stop there and just thank the people who've been involved in some of the more recent work that I described today. Frederick Lanner and Yojiro Yamanaka, who now has his own lab at McGill, have been actively involved in the primitive endoderm epiblast situation. Peter, Brian, Rob, and Katie have been looking at the early differentiation, and Brian and Peter have really been looking at the molecular pathways in the trophoblast that I didn't talk about today. So I'll stop there. Thanks.
Thank you, Janet, for a wonderful, wonderful talk. I'd like to start, we've got a few minutes for questions. I'd like to start the questions with an online streaming question. Dr. Jose Sabelli is watching us evidently.
Hello, Jose.
And he had a question for Dr. Rossant, but how do we get his question?
I can read it to you.
Okay.
Would that work?
Yeah, that's it.
When OCT4 is turned off in the human embryo, by what mechanism can bovine be a better model for human?
That sounds like a loaded question.
Yes, and since I don't work on bovine, but other people do, I think that the bovine system, it seems as though there's an even more extended period where you get this overlapping expression. The human, it's not a very long period. The bovine, it's a long period. You have to also remember, he knows, that the whole implantation process is so very different across mammalian species. And I sort of mentioned that here today with the human embryo, the trophoblast, the way it invades the whole, whole placental formation is very different. Cow is even more different. It goes much, much longer before you get any form of implantation. So I think the formation of the differentiation of the trophoblast lineage is going to be different. Whether that, and there are going to be differences obviously in regulation associated with that, because you have to, something has to give you this difference in timing. So the cow could be of importance to study that early lineage decision. I think I'm more interested, although I love trophoblast and spent most of today on it, to me the key issue for human embryonic stem cells is not so much that first decision, which has to take place, but it's the next decision that goes on within the inner cell mass. And I think we still know very little about that. And we don't know whether the cow or the human or the rabbit or the mouse is going to be the best system to study that. My feeling is, in the end, we do have to study that in the human embryo itself.
You mean the inner cell mass to endoderm?
Yeah, the epiblast endoderm differentiation.
Are there any other questions? David?
Thank you. That was a really very clear lecture. Embryo complementation has proven to be very useful in terms of testing the pluripotency of embryonic stem cells. Can your systems, or have you looked at how that occurs, how you're able to affect differentiation of the two layers artificially through this manipulation?
Oh, I've got a whole separate lecture on that, if you like.
Okay.
So with the tetraploid situation, what we know, when you make a tetraploid embryo, those embryos, if you just grow them alone...
Would you just describe that?
Oh, sorry. So in a mouse embryo, you can take a two-cell embryo and just fuse the two cells together by electric fusion, and you'll get a tetraploid embryo. Those don't make it through to term, but if you get them go through the blastocyst and early post-implantation, they're not bad at making the extraembryonic cell types, but they're very bad at making the epiblast cell types. So when you put an ES cell with it that doesn't like to make trophoblast and primitive endoderm, you get this complementation. And it's been shown that that complementation, if you do it at the morular stage, which is when we do those experiments, if you aggregate the ES cells with an 8-cell tetraploid, then those ES cells will essentially take over. Maybe I could go back, actually. So if you look at that embryo here, essentially, if you took ES cells and aggregated it here, and this is a tetraploid embryo where these cells are not going to be very good, the ES cells go in and essentially take over this compartment. They take it over at the 8-cell stage, so they're going to make these first inner cells, and then at the next stage, the embryo itself makes these secondary inner cells, Because ES cells don't like to make primitive endoderm, the secondary inner cells always end up in primitive endoderm. So you end up in this complementation situation. And we've shown more recently, was really first shown by Regeneron, and then we've looked at this in the sort of cellular details, that you can do this not just with tetraploid embryos, but if you're careful, if you take a diploid 8-cell embryo and get the proportions right, you're going to have the same effect, because you really take over the compartment at this point here, and the other ones make primitive endoderm. So it really is happening at the blastocyst stage. If, however, you inject them into a tetraploid blastocyst, then you're relying on the fact that the tetraploid cells don't like to go on to make the epiblast, and you may have some chimerism early post-implantation. So if you want to get a clean segregation all the way through, you have to do the aggregation at the 8-cell stage.
Wow.
So clear differences between a human embryo and a mouse embryo, and between human embryonic stem cells and mouse embryonic stem cells. And I wonder if you'd comment on conflicting reports about the ability of human embryonic stem cells to make trophectoderm, or not.
Yes, so it's been proposed that one of the differences, if you go over that slide, I'm not sure, I think I took it off that slide. One of the differences between human ES cells and mouse ES cells is that human ES cells quite readily in culture will make trophectoderm. If you treat them with BMP4, that they will make cells with trophectoderm markers. A recent publication, however, from Roger Peterson's lab, I think, shows quite clearly that those cells are probably not actually trophectoderm. They really are sort of more posterior extraembryonic type mesoderm. And I should point out that CDX2, which is my favorite gene in the trophectoderm, and used a lot by many people as an important trophoblast marker, is not restricted to the trophectoderm. Its name is CDX2. That's a caudal homeodomain gene. Caudal in Drosophila is involved in posterior development. There are 3 caudal genes in mammals. CDX2, when you knock it out in the embryo alone, so rescue the placenta with some of these tetraploid approaches or whatever, then you get an embryo which lacks entirely the posterior end. So it's absolutely critical for patterning the posterior development. So this is why studying human development in ES cells in terms of these early lineages is really difficult, because we cannot put them back into the embryo and ask what they really give rise to. So you're always kind of guessing. And also because we don't have enough information about the normal patterns of expression in early human embryo development. How do you know? So it looks, I don't think that those are truly trophectoderm cells.
We have time for one more question. Jesse?
I'm wondering if we know yet if the ICM PE decision can happen without the first TE inside decision. And do you have an experiment to manipulate that? Like if you make them all think they're all inside cells, will you still get the nanogeta6 mosaicity?
Yeah, so in the ROC inhibitor experiments,
right,
for example, what we see, and Robert's looking at that right now, and I can't tell you that. I don't think it's going to work. The ROC inhibitor, they get to that point, but they're not very happy after that point. So I'm not sure we can address it that way. It's an interesting question. We do know that if you take the inner cell mass out of the blastocyst, then it will, before the segregation, it will go on and segregate. But you're asking, if you really just didn't make the trophectoderm, would you be able to do that? And I'm not sure we could really do that. We've shown, years and years ago, and Tarkovsky's lab more recently with more modern markers have shown that you can take the inside cells, all these cells out and reconstitute an embryo, and it will regenerate the trophectoderm and go on and form a blastocyst. So which is what we would predict at these early stages. Everything is still labile, essentially until you get to about this point. So I would predict you should be able to do that, but I'm not sure we have an experimental system to do it.
Okay, one last.
So FGF simulation is different between the mouse and the human classes. Have you looked at the YAP signal pathway in human?
No, we have not. And that's obviously another interesting thing.